So hi, hello everyone. I hope that you're able to hear me. Welcome back to another live stream. So I'm going to quickly try to sort out all of the technical aspects first. I need to see if I'm able to hear myself. Just a second. Yes, I think the sound works. So, okay, hello and welcome to another Saturday live stream. Okay. I think that I first of all have to tell you, of course, Happy New Year. Today is December 31st, yeah, it's evening right now where I am. So in a couple of hours it's midnight, so I'm able to hear the fireworks outside. So if you hear any noise in the background, then yeah, it's just the fireworks here. Today I would like to talk about today the Microscopy home lab, and some of you sent me some pictures which I would like to show you of your Microscopy workspace. And yeah, we'll see how far we're going to get. And as always, I would like to invite you, of course, to write comments into the comments section. And if you have any questions, then of course I will try to answer them, even if they're a little bit off topic. I will try to answer them. And it's been tradition over the past couple of weeks that I always read through the comments right at the beginning, because I get greetings from all over the world. Okay. Somebody's got an adapter for the microscope. Okay, are you a microbiologist? I suppose the question is for me. Well, I did study microbiology. Yes, I got a master's degree in microbiology, research microbiology. I wrote my master's thesis in bacteriology. So I was doing biochemical analyses of bacteria, and for over 20 years I've been now working as a teacher. So I changed my profession around, and I've switched over and went into education. Okay, so there is from St. Louis someone saying hello. Hi, happy New Year. Yes, yeah. Oliver is late. Should we send him to detention? Well, well, yeah. Sometimes we send our students, I'm a teacher, they have to basically collect minutes when they're late to class, and then they have to sit it off. Okay, so I'm going to just quickly check here. Hello from Tennessee. Yeah, hi from Kashmiri. Hello from Pontefract in Yorkshire. Okay, happy New Year everyone. Okay, hello from Suhan from India. Good evening from the Netherlands. Wishing you and yours a very happy New Year. Yes, I think some of you might already be in 2023. Okay, I don't know about that. Okay, hello. Good morning, Oliver in Aussie, and having been subject to sunburn and peeling skin of my arm to get okay. Yes. Okay. Well, where I am right now, it's currently not so warm. Okay. Okay, hello from Maryland, guten Abend from Germany, guten Abend von Österreich, from Austria, from Asheville, North Carolina. You want to become a microbiologist, please help. I'm in 10th grade. Oh, if you want to become a microbiologist, then I can just tell you finish school first and study microbiology or bacteriology or molecular biology. That's a long story. Hello from England. Okay, Brazil. Okay, I collected water from 2 ponds of different parks. So many amazing creatures there. Brazil. Yes, Happy New Year. I saw your video and prepared bacterial samples at home and observed them under the microscope. Okay, from India. Okay, so hello everyone. Okay, and now let's get started. Okay, thank you for all of those comments. So what I'm going to do today is the following. I'd like to show you some of my microscopy equipment that I'm using. I would also like to share a few pictures that some of you sent me. If you still want to send me some pictures for the next live stream, then yeah, please send them to Oliver at microbehunter.com, and then I will, if you allow me, then I will also show them to the community. And another thing that I've been thinking about is if you also want to send me pictures, photographs that you made under the microscope for us to look at those pictures together, you can also do that. So my email address is oliver@microbehunter.com, and if you send me pictures, then I can share them also over the live stream. Okay, so I've got a lot of equipment here, and I will simply tell you a little bit later. We're going to go through this, and I'm going to tell you how I'm using them or not using them. But first and foremost, I would like to share with you a couple of pictures that some of my viewers sent me. me because they sent me pictures of their workspace, of their home lab at home, and I thought we're just going to look at them together. Okay, so I'm going to look at the first one here. Yeah, that's basically just a microscope here on the table, and one of the things that I kind of like with all of those pictures is it kind of shows yeah, different people like different setups, and depending on what they're interested in looking at, yeah, sometimes it's more or less elaborate. In some cases, if you only want to look at commercially prepared slides, you don't need a lab as such at all, because you're just looking at prepared slides. Yeah, so it's just kind of nice to see a little bit how different people and hobbyists, how they are arranging their workspace at home. Yeah, there's another one over here. Okay, one of the things that I generally recommend, and I guess many of you already have this, is because there's been a question, I mean, should you put the microscope away or should you keep it on the table? And I generally always say, if you have the space, then please do keep it on the table, simply because the microscope is much more accessible. And then if you want to look at something, then you don't have to carry the microscope around, and it's simply much more accessible. And then you end up also using the microscope a little bit more often. Okay, so yeah, so these are basically two pictures. So this is also one of the first pictures that I got sent. I kind of like this one here because this picture was taken while I was doing my previous live stream. Okay, so you see that sometimes people have a computer next to their microscope. This is of course then necessary if you have a camera, a digital USB camera connected. And in some cases, yeah, you don't want to have, some people don't want to have a computer next to the microscope. So for example, in my case, I want to generally do microscopy with the computer switched off, and when I take pictures, I take the pictures directly into a memory chip, into the chip of the camera, without a computer. Okay, that's simply my personal preference, so that I can concentrate more on microscopy. But then again, of course, when I'm doing live streams like this, I have to have the camera connected, obviously. Okay, so what I'm going to do now is I'm going to again go through some of the comments here again, because the comments start coming in here. Okay, from Brazil, Happy New Year. Okay, hello from Mojave Desert, California. Yes, from Romania, from Canada, from Mexico. Frohes neues Jahr, Oliver. Yes, hello. From India. Is the computer off to avoid vibrations during photos? And the thing is the following. What I have is, okay, that's an interesting point. Generally, if you have the computer connected to the camera, and if you're able to control the camera over the computer, then this is actually quite much better, because then you can avoid vibrations. You don't have to press the camera button. Okay. Okay. I also send my home lab photos. Did you check? I hope that I got it. If I happen to forget a picture or overlook it, then please apologies, tell me, and then I'll put them in next time, okay? Do you have book recommendations for microbiology? Microbiology in general, yes, I do have a book recommendation. It's not an identification book, but it's the book from Brock and Madigan. This is a standard microbiology book. I don't have a copy with me. Brock and Madigan are the two authors, and it's I don't know how many editions they already have, but that's a standard microbiology book. I have one doubt. It said that an amoeba performs multiple fissions under unfavorable conditions. I wanted to know what exactly are such conditions. Short answer, I guess it depends on the species. There are so many different types of amoeba around there that I think it is difficult. Yeah, it's difficult to actually say that. I sent it on Instagram. I did get 2 Instagram pictures. These 2 here, I don't know if these are yours. Okay. Otherwise, I could not see any others. But if they are not those, then please tell me, and I will try to get them for next time. Okay. Hello from Spain. Okay. Happy New Year. So yeah. So we see over here, yeah. And the next one over here, this is a pretty elaborate one. Plenty of microscopes, and in the background over there, yeah, we can also see that there is indeed, yeah, here we've got the arrow. Just a second, let me put the arrow over here. You see also a little telescope in the back. I happen to have the same one of them, yeah, and an assortment of yeah, regular under quotation mark compound microscopes, and also stereo microscopes. Yeah, so this way basically you can observe quite a few different different specimens. Okay, here that's another pretty elaborate setup. And what we have here is the following, that I've seen that over here there are, is also a microphone over there. So this kind of, I think, you've got some lighting here. So I guess maybe whoever sent it to me maybe is also doing some kind of streaming and online videos maybe. Some nice microscopes. I think the one over here might be a Zeiss microscope over here. From the logo, it might appear to be a Zeiss microscope. And here, another interesting setup here. Nice table as well. And over here, this here is now my room. Okay, that's in my cellar. This is where I'm doing all of my videos. So I set up this room a couple of months ago. And I put it, this is actually simply the board, the workspace of a kitchen table over here, IKEA furniture, and I simply put it up here, and over here is a little stand for the mobile phone, so that I can record myself. So if you've been watching some of my other videos on this channel, then basically you're pretty familiar with the setup. Over here I have got some cupboards where I keep also some microscopy stuff as well. Okay. So I'm going to Brock. Microbiology is the standard in Latin America as well. Brock, exactly. Thank you for writing it here. So that's the one that I've been using also during my microbiology studies. Any reference recommendations for keeping pond microbes alive for extended days or weeks? Yes, and I'm actually prepared something here. I will do that. But you know what I'm going to do? I just see over here that there is I'm going to stand up for 5 seconds, just a second. I just pushed a white little basket away. So basically this is now my little lab, or if you want to say my little studio. And this is now also the place where I am sitting right now. Okay, and this is where I'm doing my live streams. Okay, I've got a 3D printer here. This is a little camera with a teleprompter for doing my videos, primarily for my other YouTube channel. I've got several YouTube channels which are microscopy related. And and here that's basically the microphone over here, which you cannot see right now because it's kind of below here. Okay, so you're not able to see this. And over here I've got a camera, a little webcam, and this webcam up here is now basically recording my face. Then this little camera over here is recording the surface. Basically the tabletop that I just can show you. I can actually hold it up here as well here. It's a little document camera that's also used for recording things, documents and paper on the table. And I kind of like this one because it allows me to focus it as well, so the lens can be focused. And over here in this plastic container on the side, this is where I keep my microscopy tools. Of course, I've got a cupboard as well with drawers, but simply to make it accessible, I simply put them here into a box. Of course, tissue paper is always necessary as well. Okay, and one of my cupboards, this is how it looks like. A whole bunch of chemicals that are more or less often used containers. Those graduated cylinders I got once, but I'm rarely using. This centrifuge, some of you might already still remember. I've got some immersion oil over here. Over here in the background, some mounting media and some solvents. Over here I've been experimenting around with different types of inks and stains. Okay, and there's some alcohol, some acetone, and some solvents that I'm occasionally using. Okay. I was the first who sent firstly my photo. Okay, hi from Yorkshire, England. Okay, so I hope that One Tap Gamer, I hope that I got your pictures. Otherwise, if not, I apologize and I will try to look for it. Okay, so this here is also in my cellar. And over here, I just want to talk a little bit about this microscope over here. This is a very old Olympus CHA microscope. From the 1980s, early 1980s. I got it secondhand, and when I got it, it was not functional at all because the focusing system and also the mechanical stage didn't move anymore at all because the lubrication oil started to solidify. This is something that happens quite commonly when you basically don't clean the microscope for a long time, the oil, the lubrication oil, will start to become solid. So very difficult to turn. And if this happens, you should not force turn it. So I sent this microscope here to a servicing company who is specialized, and they're actually authorized by Olympus. And yeah, I got it serviced, and they completely took it apart, cleaned everything, and now it's basically as new. Yeah? And down here, a small stereo microscope. Okay. Okay, the second photo is his. I love your channel. You have given me great ideas on setting up my new hobby lab. Okay, Happy New Year. Okay. And then over here, yeah, that's another, yeah, also in the cellar, two other microscopes that I've got. This one is an introductory microscope, and of course also another stereo microscope. And this little device here on the side that you see, that's an interesting one. Yeah, maybe I'm going to show it to you again. I got this also second-hand. It's an interesting one because there is a little mirror in there, and you could put it up over your photo tube, and in the front here there is a projection screen. So actually this is a way that you can actually project the image of the microscope on a glass surface in front. It's yeah, totally obsolete these days because you connect the camera, but it's kind of interesting to see anyway. Okay. Okay, a few more questions. Were you interested in microscopy as a child? I got my first microscope when I was 6 or 7 years old, so I was of course always interested in the sciences. But my first microscope, it was okay. It was a toy microscope, you know, not a lot to expect there. But the problem at that time is especially when you're a child, and if you want to use a microscope, you need to know a little bit about sample preparation. You need to know what to look at, and I didn't know that, obviously. Yeah. So I just looked at the slides that came with the microscope, and that's pretty much it. Yeah? And then, yeah, sooner or later you start to lose interest. And only later on, when I got older, I basically got more serious about microscopy, and then it was, of course, also more experience, and I could make my own specimens as well. Okay. Which microscope do you use the most at this moment? The microscope that I use most currently is my Olympus microscope. It's a BX53 microscope. Yeah, which is quite a nice one that I have. Yeah, but I use all sorts of different microscopes depending on what I want to look at. Of course, if I want to do stereo microscopy, then I of course use stereo microscopes. Yeah. Any microscope recommendations? This is a very, very common question, and it's a very difficult to answer question. And the reason why it's so difficult to answer is because so many different people have different expectations. Okay, and for this reason, yeah, it much depends on money. Yeah, and generally what I would say is the best microscope is the one that you end up using the most often. But generally I recommend not to start off too expensive. Don't buy a toy microscope, I would say, but start relatively low cost. Learn as you yeah, go along, and then you can always make a more well-based decision later on if you have more experience. So I'm not recommending any specific brand or model, or I'm also not recommending one because I think there are too many different yeah, variables here, but rather I think what I recommend is learn as you do the hobby and start to build up on. Okay, can you show some paramecium under polarized light? Yes, I can do that, and as a matter of fact I have got some samples here, and I wanted to do this anyway because otherwise I would not be able to show you anything today if I just show you the workspace and all of the tools, and I still want to show you something under the microscope. Okay, so I will do this as well. And what I would like, just a second, and I turn on now my microscope. Okay, here I'm back again. Okay, so I'll do this later. But what I would like to do first is I would like to show you some of the tools and how I use them or how not use them, and give you some recommendations here. And yeah, so there was already one question, and this one question was the following: how do you keep slides with water microbes? How do you keep them alive? Well, of course, just a second. If you have, of course, a water sample, like I have over here, there are a whole bunch of paramecia in here. Well, of course, you can almost keep it indefinitely. Water will evaporate, and then you add a little bit more water. Do not close it off. This is really important. If you close it off and if you put a lid on it, it's going to go anaerobic, and it's going to turn bad, and everything's going to die off. So keep it open. But the question was now, once you have already made a slide, how do you keep the slide, the organisms alive on the slide? And what I've done is the following. I 3D printed myself this little thing here. You don't have to 3D print it. You can use any other system as well that you want. Maybe you can make yourself some kind of a rack. And the idea is the following. This is a so-called a moist chamber. This is a simple plastic box for lunch and for food. And you put it in here, and you put a little bit of water down here. And the rack will basically prevent the slide from actually touching the water. So if you've got a slide in here, you just put the slide over here on top. Okay, there is water on the bottom, and then you put the lid on top. And this basically means that the slide is not able to dry out, and you can keep it for a very, very long time. Okay. As a matter of fact, there is even the danger a little bit that water is going to condense here on the lid and going to actually drip down. So it's definitely moist enough. And this is a way that I actually keep my slides, okay? This is actually quite a nice and convenient way. If you don't have a rack like this, what you can also do is you can put some tissue paper in here, and then put some water into it, and you put the slide on top of the tissue paper. This also works, okay? Or I don't know, maybe you can come up with some other system where you can have a little spacer so that you can put the slides in here so that the slides do not become flooded by the water. Okay, but this is actually something that I recommend. Yes, I also made some time ago a video on this, but simply wanted to quickly show it to you. Okay, so there are a couple of more questions. Can you show us some paramecia? My home lab is in Thailand. High temperature over 30 typically, and high humidity. I have aircon when I'm in the lab only. Is it better to have no aircon because of changes in temperature and humidity? That's a difficult question. Okay, so especially in the tropical areas, what I've heard is there can be a problem occasionally with fungus growing on the optics of the microscopes. This is, as a matter of fact, a fairly common problem. How common is it? It's so common that some microscope manufacturers actually advertise optics that are coated with an antifungal coating. So that it reduces the growth of fungi, especially on the eyepieces and so on. And this is because of the high humidity. Now, the problem with air conditioning, as far as what I know, is indeed that the temperature changes that you have, they can cause moisture to condense on the optical surfaces. So it's basically cold, and then you turn the aircon off, and then it becomes warm, and then you open the door and moisture comes in, and it starts to condense on the optical surfaces. That can be a problem. However, what I also heard is that air conditioners remove moisture as well. So I guess it's some kind of a trade-off effect. So what I would generally recommend is, and maybe some of you know a little bit more about this, is maybe the temperature changes are not so much of a problem, but make sure that the humidity is kept generally low. So that means if the aircon is off, keep the doors closed so that the humidity is not able to enter the room anymore. Okay, so this is simply a recommendation that I have here. Yeah. Recently, do-it-yourself gene editing has become very popular with the help of CRISPR-Cas9. What are your thoughts on it? Will you try it? No, I'm not going to try it. I don't know about, honestly, do-it-yourself gene editing. I mean, I did a lot of DNA work when I was at university a long time ago. I don't know. I mean, at that time, what they sold is they sold a whole bunch of kits for labs. Okay, so we were called the kit kids because we basically got all of the DNA and all of the enzymes and everything prepared. But I don't think that this is actually designed for home use. Okay, I guess these are some kind of kits that the company sells to make actually working in the laboratory easier. Okay. Yeah. I also made this rack with popsicle sticks. Yep, that's a good idea. Okay, I'm keeping a single drop of water full of algae inside a moist chamber since last week. Yep, that's also possible. Where do you get your lab materials? Directly get it from science store that nearly my house. So where did I get my lab material? So all of those tools here, this one over here, the scissors, this one, all of those here, as a matter of fact. Okay. those 4, I think. Okay, not the others here. They are almost, I don't know, over 25 years old already. I got them back at university when I did my first dissection course, and I'm still using them. You know, so they're actually pretty good quality. We're not that expensive, and you can buy stuff like this over Amazon. There are really a lot of products on Amazon, and what you have to do is you have to search for dissection kits, because there are lots of med students and I don't know, who are buying those dissection kits. And however, what I've seen is that they contain many more tools than actually necessary. So I think the only important tools here are those tweezers over here. I use them all the time to pick up algae and samples, even water drops. And those scissors over here are very useful because especially when you pull out some filamentous algae, then you can cut it off using those scissors. So I think of all tools, these 2 I'm using most often. Occasionally I'm also using this here. This here are tweezers actually for stamp collectors, but sometimes they are quite useful for picking up insects and other small animals, because they're not quite as pointed, so you cannot do as much damage to the insects as if you were to use those here. But actually for insects you actually have even softer ones. Even that one is actually quite strong. Okay, so and occasionally I also use those, but not so often. And sometimes the stronger scissors are also quite useful, especially if you want to cut through some harder plant material to make it smaller a little bit. Okay, so these are actually from the I would say the core set of tools that I'm using, and I would say that there is nothing special about those, and you can get them on Amazon and pretty much from any a lot of shops. Okay, I'm going to look again at some of the comments. Where do you get your lab materials? I directly get them from a science store. Yep, that's possible. Amazon is the one that I'm using. Do you have an electron microscope? No, I don't have an electron microscope, and I do not intend to get one because I cannot afford it. But that would be the least issue, I would say. The bigger problem with electron microscopes is that it takes days to prepare a specimen. Okay, it takes days because you have to completely dehydrate the specimen. So it has to be completely free of water. Then you have to put it into a vacuum chamber, and you have to coat it with a single atom layer of metal to make the electrons reflect from the surface. So that's an elaborate one. I've seen electron microscopes while I was at university. They had a few of them there, of course, and we did some tours, but you had to pass an exam and a course before they even let you near the electron microscope. And often it was like this, that as a student you were not allowed to work with them anyway, and also other researchers were not working with them. Essentially, if you had something to be observed under the electron microscope, you had to give the specimen to the person who was in charge of the electron microscope, and he or she did the microscopy for you. So it was kind of like, they were the experts, and you just gave them the sample and they sent you the picture. So this is kind of the way that it worked. They are for home lab only, just to experiment at basic level. They give E. coli with kids. Oh, that's about the CRISPR-Cas9. And with the help of the kit, we can make the bacterium resistant to some type of antibiotic. That's interesting, actually, to know. Interesting to know. I didn't know that. This would be kind of interesting in the sense that because I'm a science teacher as well, it would be quite interesting also to use in school. This was, I think, this home lab CRISPR-Cas9. Do you have any? But then again, it depends a little bit on the legal situation. You have to understand that not all countries allow you to do genetic modification at home, because they're kind of worried that if you change the DNA around, that those organisms actually become released into nature and into the environment, and they're not always happy. So I remember that when when we did our first DNA modifications at university on plants, we did an experiment on plants. They were actually counting the individual plants and that were modified, and we had to return them individually, and they had to be officially destroyed. So there is no accidental release of genetically modified plants into the environment. The laws were quite strict about this. Do you have a recommendation for a 3D printer? I must have one to make things as you have shown. I would love one to stand up to UV light the sun for some outside water projects. I don't have, well, again here the 3D printer, there are so many of them around. The one that I have is the Ender 3. It's a very common one, was not so expensive, but I would say if you want to get yourself a 3D printer, it's only worth it if you actually spend some time with it, learning how to use it, because it was not so straightforward as I was kind of hoped it to be. Yeah. So, but now that I kind of worked myself into working of a 3D printer, I'm also able to make simple things now. But it was not like this that it kind of I was able to make things directly out of the box. Okay. So yes, back to this one over here. Important tools over here. So a couple of containers that I'm using to collect specimens. Okay, because occasionally you're going somewhere and you don't know whether you're going to see some interesting specimens and you want to take them along. So what I do is I got myself those plastic containers. These are centrifuging containers. So basically they're disposable ones. So you can get them in packs of, I don't know, 50, 100. Amazon again. Yeah, but these are used very often in labs. They have a screw cap, as you can see here, and the nice thing, they are watertight. This is really nice. Okay, so you can use them for water samples, and not only water samples, but you can also store other things. And I like it, of course, because they are transparent and therefore you know what's in it. Okay, so usually when I go somewhere, I always take a couple of these containers along, and if I find something, I simply put them in there and take them home, and then I put it under the microscope. Yeah, I also had those here. Yeah, I don't like those so much, the clear ones, because the plastic is relatively hard. I mean, it's more clear and more transparent than the other one here. However, it's also more brittle. Okay, so yeah, the others over here, they are a little bit softer, actually significantly softer, okay, and therefore they don't break quite as easily, and yeah, so I actually recommend those here, and they're quite common, extremely common, and you just search for centrifuge tubes. This one is 15 milliliters, and this one here is 50 milliliters, okay, so this is kind of the thing that I kind of recommend, yeah. So yeah, the acrylic Viper is a nice 3D printer. Okay, yeah, it's fine. Okay, where can you get a high concentration of amoeba? You always have to be careful when you're growing your own microorganisms. Okay, but what I have done, I also made videos of this. It's kind of difficult to find them because I've already made so many videos. If you go back a little bit, what you do is you take a water sample like this here. Okay, I put some aquarium plants in here, and I added a cereal and as a food source. And then when this happens, you start to multiply paramecia like crazy, but also amoeba. Yeah, so there are plenty of amoeba in there as well. They can be a little bit more difficult to see than the paramecia because they move much slower. Okay, and the paramecia and other ciliates, of course, they move around very quickly, sometimes a little bit too fast, easy to see, but the amoeba themselves, because they crawl around, yeah, they're not quite as evident, and you have to be a little bit, you have to look for them a little bit more, okay? So I got my first microscope when I was 8 years old. Now I'm 56 and still have the toy Sears microscope. Yep. Yeah, sometimes the first microscopes, there's a certain nostalgia attached to it. So that's why. Yeah. I know that on a number of images you present, a blue filter is being used. Okay. I'll talk about this. No, it's not a blue filter. I'm using a DIC microscopy. And however, in one of my previous videos, I also showed this. Just a second. You can use a blue filter as well. Just a second, I gotta reach down into my drawer. Look what I got here. Okay, this here is just a clear plastic, and I put some blue tape over it, and I made a little cutout. So this means that there is now white light striking the specimen from the side, and the background is blue. So you can experiment around with these things. And just a second. Yep, here 3D printed. Yeah, so this is like a dark field filter, and you don't have to 3D print it, but you can use some kind of clear plastic and use some blue marker pen, And if you use a filter like this, what's going to happen is that the background is going to be blue and the specimen is going to be bright, okay, because the light will strike the specimen from the side. Well, why am I holding it up here? I can actually show it to you down here as well, okay. So I've been using a 3D printer as well to make those, and you can control the intensity of the blueness by controlling the thickness of this. Yeah? So you can get a very similar effect, but in my case I did not use those filters. Okay, I've received over the last couple of days several of these questions about the blue background. Okay, hi from South Africa. I bought a ringing table and camera after your last stream. Wow. And I'm now practicing creating slides. Thank you for all of your knowledge. Okay, you're welcome. Okay, currently I'm using an ATA photographic filter. Ah, interesting. Could you provide reference or recommendation for stains used on protozoa? Okay, staining. It depends what you want to stain, number one. And for protozoa, specifically for paramecia, there is the so-called, I've never used it, okay? I just know it from literature, the so-called the silver stain. And the silver staining method is actually used to make the cilia visible, and this is used for identification and classification purposes. So if you're specifically interested in staining protozoa and paramecia and other ciliates to make the little hair, the cilia visible, it's the so-called the silver stain method, but be aware that all of those staining methods will of course kill the specimens. Okay, so for this reason, live staining is difficult to do. Okay, because many of the stains, well, if they bind to certain parts of the cell, they will of course then kill the cell. Right? So you can only use them in very low concentrations. But if you want to use a general purpose stain that stains a lot of things, then I recommend methylene blue, also known as Löffler's solution, because this indiscriminately stains specimens. Yeah? But because it contains alcohol and potassium hydroxide and so on, it's going to kill the cells. Okay, so live staining is difficult. Okay, I'm just saying, but there are a whole, depending on what you want to stain, there are of course different protocols here. Okay, no, I did not, but it did not work. Even I started microscopy 2 years ago, but did not see amoeba. Okay, how is it going? I suppose I could construct an airtight case for the microscope to keep the humidity away while not in use. What you can try to do, for those of you just joined, there was the question about humidity and storing of, and in a home lab situation when there's moisture. Maybe you want to try silica gel. Silica gel, these are the things that are sometimes included, those little packets that are sometimes included with clothing and other things, because they kind of absorb the moisture. You might be successful with that. Okay, and another thing that I might recommend, again, I've not used it, I've not tried it, but if you use baby diapers. If you use baby diapers and if you cut them open, there's this white powder in them, and this white powder is highly absorbent. You take out the powder of the baby diaper, put it into a glass and add a little bit of water, and it really absorbs the water and starts to swell up. It's really amazing. And so I can imagine that this powder in baby diapers is also quite moisture absorbing. So you might want to try this as well. I have not tried it. Okay, I don't know if it works, but this is simply something where you might want to do a little bit of more research in keeping the moisture down. Okay. Can you teach us how to use a pipette? Okay, well, there are pipettes, graduated pipettes, and there are those simple pipettes like the ones over here. They're pretty easy to use, really. I'm going to talk about this here a little bit because what I like to do is I would like to show you those three here. Okay, maybe you are actually referring to those pipettes, the microliter pipettes, because they actually require a little bit of not difficult to use, but you need to know how to use this. Okay. So I'm going to simply do that. So I recently bought myself a so-called a microliter pipette like the one over here, and normally they are extremely expensive. We're talking about here several hundred euros, several $100s. And not affordable, and I would say also not meaningful and not necessary. And the reason why they are so expensive is because they're calibrated, because they're used in molecular biology and you really need to be very precise. Now this one over here was ridiculously cheap. I think it was only between $30 and $40, 30 and 40 euros on Amazon, okay? And it also works. I do not need the precision and it does not have to be calibrated. And again, I bought it myself. It's not sponsored or anything, so it's just happened to be the cheapest one, okay? And those pipettes have, a scale over here. And this one over here goes from 100 microliters to 1000 microliters. So that's a milliliter. And the milliliter is, if you're not used to the metric system, it's a 1 centimeter by 1 centimeter by 1 centimeter, okay? And you can change now the volume that it picks up, okay, by turning this one over here, yeah? So if I want to go all the way up, okay, to the maximum, I have to turn this all the way to 1000. Okay, so I'm now at 1000, okay? So that's now basically the maximum capacity, a milliliter, okay? And and how do you, I don't need so much. So how do you actually now use this? Okay, it has basically 2 buttons, okay? This one over here, okay? And you can also continue to press it, okay? So there's a first stop and then there's a second stop, that's important. And then there is a button over here. Okay, this is the eject button to kind of throw off the tip. So how does this work? As a matter of fact, I consider these, if you really want to spend a little bit of money, I consider them really practical as well. Okay, so I like using this because it's really nice in the hand. Not absolutely necessary, but it's a nice luxury item. Okay, and it looks kind of cool. So you buy yourself a box of disposable pipette tips. Okay, and then you do the following. You put it in like this. Okay. And you take it out, okay, and here we go, okay. So, and if they're disposable, so this means if you don't need them again, you press this button over here, okay, and then if you press it, then it's going to kind of eject it. And because this is a bad quality pipette, it's not bad quality, but it's kind of, it could have been made a little bit better. I think sometimes it also goes off, so you have to put it back in again. And get stuck sometimes a little bit. See, that's the difference between a 30 euro and a 300 euro pipette. Okay, okay. Ah, did I now really manage to break it? Okay. Okay, wow. Ah, this has to go in here. This little thingy here has to go in here. Okay, here we go. Now it works again. Yeah, and how do you now pick up water? I will show this to you. I need some water. Yeah, I also recommend this here, a little water bottle, because I do not have running water where I am. And I always need a little bit of water for my samples and specimens. So I like to use this one here. And let's add a little bit of water in here, simply for demonstration purposes. And what you do is now I have to kind of, I've got, there's a microphone in the way here. So, and what you do is the following. Yeah, you basically press it, okay? You go in here, okay? And then you release. And when you do that, you're taking up exactly the same volume as you indicated here. So this one is now 600 microliters, okay? Yeah, and then, and then if you want to get it out again, then all you do is you kind of push it, obviously. Yeah, so you push it all the way in, and you see that there is still a little bit of water left here on the tip. And this is now where you have to push it all the way in, and this kind of pushes out the remaining water as well. Okay, so for pulling it in, for drawing the water in, you just go to the first stop. You basically draw it in. Okay, and for pushing it out, I'll show you on the top, you go all the way out. Okay, I'm going to show you a little bit how this actually looks over here again. Yeah, you do it like this, and then when you press all the way, then it kind of spits out the remaining droplet as well. Yeah, and then when you're kind of finished with that, and if you want to throw the tip away, just press this button here, and then it shoots out the tip, and then you can basically put on a new one. I don't throw them away, obviously, because if I just use water, then it's going to be fine. They're not sterile anyway. They don't have to be sterile. And in a microbiology and genetics lab, they have to be sterile. So what you do is you basically, you're not allowed to touch them at all. And you basically make sure that the whole box is sterile by autoclaving the whole box, by basically putting it into high heat, okay, steam. Yeah? It's not necessary for our purposes because we're not growing any bacteria or anything, and it's not sterile anyway. Yeah. So this is kind of the way that this pipette works, and I generally just leave it at one setting. Yeah. And I do not use it for measuring out any volumes. I just use it because it's a very convenient way of actually picking up specimens and putting it on a microscope slide. Okay, so I'm going to read off a couple of questions again. Okay, there are a lot of comments now. Okay, I think about hello. I'm just a second. I've got to scroll up here. Use a pipette. I just found an amoeba after 2 years. I'm recording 3 paramecia. Okay, the variable pipette. Yeah, that's called a microliter pipette. Yeah. Are there any general statements regarding staining proteins, chitins, for example, polar compounds for X and basic non-polar stains for Y types of compounds? Yes, there are. I forgot the stains. I did a lot of staining in during this was over 25 years ago. There are stains for proteins and for all different. Let me put the cable away. For all different substances, but I don't know them now off my head. I should look for papers or books on staining. Yes, I would recommend that. But be aware that some of the stains are not healthy, okay? Because if they are actually able to stain certain parts in microorganisms, then of course these are cells, and then they are also able to stain your cells. So you got to be careful here because they react with your own cells as well. So you got to be careful that they're not. So it's not dangerous, okay? And some of the companies might not sell all of them to private individuals. Just saying, okay? Could use dehumidifier packs for car caravan pretty cheap and can be reused by micro. That's interesting. So this was in reference again to the humidity in some of the home labs. Okay. Hello, I'm an extreme macro photography where I use tubes and objectives mounted to the camera. In some time I managed to get a few decent objectives. Okay. Yes, some people mount the objectives directly, microscope objectives even directly to a camera. And I never tried this, but I know that even some people were 3D printing some of those tubes for their camera. So at the front, they are adding an objective, and then they have a 3D printed tube that they can directly mount to their DSLR camera. I'm thinking about moving further with my hobby and considering a microscope. Would you recommend to reuse these objectives? Also, they have M26 thread, while most microscopes RMS thread. Difficult to say. You see, it depends a lot. If you want to get into into upgrading and reusing and microscope maintenance, if this is your interest, then why not? Okay, it might take you a little bit longer to actually put together a microscope yourself, but you learn a lot in the process, and if you enjoy doing that, then why not? If you say, okay, I'm more interested in just in the observation part or in specimen preparation and so on, and I'm not so much interested in upgrading and in the technical aspects, and then maybe not. Yeah? So I would say this depends a lot on your personal preference and into which direction you want to develop yourself. I know nothing about microscopes. Are there any adapters I can mount my M26 objectives to the RMS revolver? I don't know. Okay, honestly. This is something where I'm sorry, I got to say I don't know about these adapters, whether they exist or not. Okay, Happy New Year. Yes, thank you. Why sometimes we see protozoa in couples are glued together? Are they sexually reproducing? Yes, there can be 2 reasons for this. Sometimes you see 2 protozoa together. They could either be conjugating. This means they're fusing together to exchange DNA. Or, so basically if they are longitudinally, and if they're the other direction, then probably they're dividing. Okay, so it depends what they're doing. Yeah, it depends a little bit on the direction. So they could be either doing mitosis, regular cell division, or they could be actually conjugating. That means they're fusing together, exchanging DNA, and then they're separating again. Okay, to dispose these tips or clean them? Well, very simple. if because if I only use water, I of course always reuse them. Okay, but in a microbiology lab, there is no reusing, because once contaminated, it's contaminated. It goes away. It's thrown away, right? But in our cases, it's not so important. So, but what I don't do is, I mean, it's also not a big deal. But when I, for example, have a water sample over here, and I put some paramecia in here under the microscope slide, and then I would like to observe a different water sample, I'm not using the same tip. Okay, because if I were to use the same tip, I'd be transferring specimens from one glass to the other one. Yeah, and then basically maybe have different, then they're going to start reproducing there. I mean, also not a big problem or an issue really, but I want to keep my specimens kind of separate, and and that's the reason why I do not use the same tip for different water samples. Yeah, because I just want to keep it separate. But then again, it's not a big deal, because I'm just interested in observing what I can find. Yeah, and sometimes if I start to transfer now some organisms over and they start reproducing in another container, yeah, who cares? Yeah, but I just don't do that. Yeah. So for this reason, it depends a little bit of what I've used. Or for example, if I've been using it for some salt water or sugar water that I've been pipetting, then I'd simply rinse it out and I save them, because why make plastic waste? Yeah. So, but it depends a little bit. Sometimes they're so dirty, I just throw them away. By the way, I just want to show you something else. I want to tell you a little bit here. The tip at the top is quite small. It's still big enough for cells to go through, right? But what I found is that because it's still too small, and when you suck in the liquid very quickly, especially for water fleas, this is too small. Okay, and water fleas and Daphnia and Cyclops, they're pretty big really, right? And if I start to try to suck it in over here, they might become damaged, right? So what you can do is the following. This is where these large scissors become valuable again. Yeah, you simply do something like this. You simply cut off the tip, okay? And now it's large enough to pick up larger organisms. As well, yeah. Just a short recommendation, yeah. Can berry juices be used as a non-lethal stain? Pokeberry. What I think about berry juices, you can try it out, but I'm not sure whether they're actually strong enough. Okay, I mean, what you usually use for staining, like methylene blue or even fountain pen ink, and so on. I mean, these are pretty strong stains. If you get some of the, so I think it's got to really stain your fingers as well very strongly, and then I think it might also be strong enough to stain them. But again, it depends quite a lot on the actual chemical composition as well. Yeah. Can you please tell me what the solvent we would recommend to clean objectives? I have a second hand, don't have access to this manual. Okay, I don't know these objectives generally, but there is a general recommendation, and that I've heard, and that is you use a mixture of ether and alcohol. It's very volatile. This means you do not use any solvents for cleaning that are used for eyeglasses or so, but you use highly volatile solvents, so they evaporate very quickly. And generally, regular alcohol is recommended, but make sure that you are not using too much. Otherwise, and you use. I mean, I don't have an objective here right now. It's in the cellar. You know what? I'm going to illustrate this here. Okay, so let's say let's say that this is our objective. Okay, so this is our objective, and you take some tissue paper, and what you do is the following, and this is the lens, okay, and you put the tissue paper over the lens like this, and you add a drop of alcohol on the tissue paper, and then you pull it over like this, okay. So with very low pressure, you or almost no pressure at all, you simply let the solvent do the action, okay. And this is how you clean the lens of an objective. And because it's so volatile, it's going to evaporate very quickly. Now, however, I was told the following: because the objective lens, the front lens, sometimes is concave, it goes inwards a little bit, you might not get all of the dirt out that's in there because it goes inwards. So the service person from my company, Olympus company, told me the following: don't be afraid, he said. You can actually, don't use a sharp object, but you can actually use your fingernail and carefully actually try to remove any dirt that's in there, in case you got some dirt in there as well. Yeah. So he said this is that sometimes those objectives are actually able to withstand more than what you might think. And he said sometimes the people are a little bit too careful, but you won't get all of the dirt out sometimes. Yeah. So I'm just saying that what I was told, and I can only tell you, whatever it is, use highly volatile substances. Volatile means it's to evaporate very quickly, because you do not want that those solvents actually have a long time to interact with the lens, and possible some lens cement or lens glue that was used to hold the lens in place. Okay. But specifically, I do not know about those objectives. Would it be great if you can link to your videos? I watched a few of the subjects and you mentioned methanol with not methanol, ethanol. Methanol is poisonous. You don't want to mess around with methanol. Ethanol is the regular alcohol. Okay. But I never purchased any and was not sure which exactly it should be. Well, it's a mixture that I got from the drugstore. I think it was something like, in a book I read, was about 70% ether and 30% ethanol. Yeah, so something like that. Yeah, I've had a good experience with Joan Lab. I own one of their centrifuges in the heat block. Okay. The best microscope for $100. There are lots of introductory microscopes for that. Yeah, So difficult to say. What kind of heat block are you using? I'm not using any heat block or heat plate. Okay. I don't need it, but I know that some people might need it to make sure that sometimes when you make permanent slides, that it evaporates faster. Okay. Is there a small 18-well heat block? I think it's 18 well. I'm not at home right now. Okay. Yeah. I'm doing non-toxic DNA extraction study on culinary vegetables. DNA extraction, yes. It's. Is there a YouTube channel specializing in objectives connecting to cameras with a bellow or tubes? I don't know about that, but you might actually find some maybe in macro. Maybe there's some macro photography channels out there or some forums. Maybe they are able to help you. Okay. Pressure cooker to sterilize, yes. Yeah, I have held up well. Alan Wells Photography YouTube. Okay, yes. Show other things of your home lab like stain, chemicals, glass, and so on. Okay, regarding the YouTube channel you mentioned, sure, I followed too. I managed to build reliable setup. Okay, I'm going. There are lots of comments, which is really great, but it takes me a little bit of time. Okay, the problem with this you mentioned channel is that it is extreme macro, not microscopy. Yes. What about coatings on the objectives lenses? Yes. Coatings is that's the whole thing. That's why you have to be careful with which type of solvent you're using. But because alcohol has been recommended by some manufacturers to clean the lens, then I think that's what we should go with. On the other hand, I have to say, generally there is no reason to clean objective lenses unless they become dirty. But why should they become dirty? The only reason why they might become dirty or contaminated is with immersion oil. And that should only be the 100 times oil immersion objective. Okay, and that you do not need to clean with a solvent. You just use lens paper to remove the immersion oil. Otherwise, there should actually not be a reason to clean the objective because it should not become dirty. Yeah. So I'm just saying only clean the things if it's really necessary. Okay. Is it important to specify if it's diethyl ether or petrol ether? I think it's I don't know about that. It's just regular ether. I think it must be diethyl ether, I guess. It's not petrol ether. Okay, yes, diethyl ether. I have a question. What magnification can I see white blood cells? Okay, I'm making a white blood cell video. You can see white blood cells easily already with total magnification of 400 times. But any higher magnification will, of course, make it easier to see. Okay. On the CD Fuge, yes, I know. I've been promising you to make a CD Fuge. I'm planning on this. It's a centrifuge using a CD, okay, which you can spin by pulling apart two strings. Okay. So you know what? I'm quickly more comments are dropping in here. Okay, I'm experimenting with used food coloring. Yes, you can use food coloring as well, especially Easter egg colors might work if the Easter egg colors are in powdered form. The red one might be carmine. If you have carmine, just check whether it contains carmine. Then this is a common stain that is used in microscopy also to stain chromosomes. Okay, so let's move on a little bit here. Look, this one is my favorite one over here, and I have not found it anywhere else. I don't know where to buy those things here. These are very shallow dishes, glass dishes, and there are staining dishes, and they're extremely useful, I think. It's kind of fairly heavy because it's made of solid glass. Okay, it is rounded here, so it's easy to wash out and wipe out because there are no corners. Because usually if there's some corners, then it's kind of more difficult to reach the corners. I really like this thing here, but I have not found any place where they sell it. Yeah, so I'm just saying that this is especially for staining and so on. So however, I do have a recommendation here. Also, for example, if you need to keep some water. Okay, those jars, they can tip over. Of course, this one cannot tip over. But look what I've got here. I'm just going to open this right now because this might actually serve as an alternative. These are small Petri dishes. Okay, look, they're even wrapped in paper here. Okay, and I think that they can also be quite useful. And I've also been using them occasionally, to keep samples of stains, for example, or for example some water samples. Water samples have an advantage here as well because they have a large surface area, so oxygen is able to reach it much more easily. Okay, and of course you can put a lid on top here, and this is indeed also a possibility. But again, I think these are a little bit easier to clean out because they have corners here on the edges, which can be a little bit hard to reach if they become dirty. But highly recommended as well. Disposable pipettes. They can be bought in packs of 100. Same here. The tip is a little bit small to take up water crustaceans like Daphnia, water fleas. So I recommend that you cut it off. And then you can, basically quickly, push it together. You put it in, you quickly release it, and it's going to suck in, very quickly the water fleas. Okay. Also, something that I occasionally take along. I don't know the English term. We call them in German Eppendorf-Gefäß. So we call them Epis. Yeah. Eppendorf containers maybe is the ones the way that you say it in English. These are also can be used for centrifuging, and they are very useful also to carry small specimens along. So I use them to carry things home. Not very useful for long-term storage because, of course, the quantity is kind of low. That you're able to store. So if you want to keep samples, you actually want to use those containers here. But if you have some dry samples, I don't know, some occasional insects that you find somewhere and you would like to take them home, then I recommend those here as well. Okay, and then of course you have those glass pipettes, dropper pipettes, which I don't use very often really, because if they become dirty, it's kind of difficult to clean. So what I usually do is I use those disposable ones. I rinse them, of course. Then I reuse them, and if they really become dirty because some algae or some material started to dry up in there because I forgot about it, I simply throw it away. But cleaning those is actually not so easy, but they are actually quite useful because it's made of glass for actually picking up solvents, for example. Okay, if you want to maybe dilute something with a solvent, then actually they're better because plastic and solvents might not always be very compatible. Okay, Yeah, and then of course I have an assortment of microscope slides. I'm now using microscope slides of a slightly higher quality. You might not see this very well, but the corners here are not sharp. They're not 90 degrees, but they're 45 degrees. And the edges here are also not sharp. They're polished as well. Okay, so they will not chip as often. And what I'm doing is I'm actually reusing them. I'm washing them. Because very often I simply have some water samples on here that can be cleaned off very easily and rinsed off very easily. So I simply collect them in a plastic container, and then when the container is full, I simply wash them with hot water, a little bit of detergent, soap, and then I wash it individually and dry it, and I reuse them. Yeah. And concerning cover glasses, I also want to show you something that I've got here. I find them quite practical as well. Look at those huge cover glasses. Okay, they are pretty large. And the reason why I also like to use those is because when you use those, the specimens do not dry out quite as quickly under the microscope slide. Because the surface area or the edges, if you put it on here, it basically doesn't dry out as quickly compared to the small ones, because there's simply more volume beneath the cover glass. So for long-term observations, this might also be useful. And basically, it's a little bit a question of taste as well. So I'll go back to the comments now a little bit. Okay, I quickly go down very quickly. I'm reading everything, but I'm not reading out everything aloud. Okay, microtubes. Ah, yes. In the UK, we call them Eppendorf containers, vials. That's the term. Yeah. Storage pod depends on those small tubes are called vials. Ah, okay. Have you ever heard of an issue with cover slips having imperfections? Appears almost just like I bought some clearly poor quality. I'll be honest with you, the quality of cover glasses and microscope slides is an issue, and I stopped worrying about it because it's always an issue. There's no point worrying and feeling upset about it, because I felt upset about it because I bought myself some nice cover glasses and they're all dirty. It's unbelievable. Okay, so I just said forget about it. I'm just So what I usually do is the following. Pretty much all of them have some kind of a grime on it, and what I do is I got myself this microfiber cloth for eyeglasses, and I simply dry wipe them, and this actually works quite well. Okay, sometimes simply rinsing it with water does not work, because it seems to be some kind of a fat or oil or something on it, This actually works now. Actually dry wiping it is actually quite okay. Occasionally previously I've also used those, where is this? I don't have it here. Simply tissue paper, here it is. Tissue paper. But those tissue papers, they leave behind fibers. Not a big deal. But then sometimes you have fibers, under the microscope. Those cotton fibers and not cotton, cellulose fibers. And with those microfiber cloth, it doesn't do that. Can you please tell me what you think about the use of strong electrical air blowers to clean the computers, to clean objectives? Can a strong one damage objectives glass somehow? I don't know how strong they are, but what I'm doing, I don't have this here. What I've done is the following. Don't use those compressed air blowers that contain this, there's sometimes this liquid in there that you use for computers and keyboards. You can buy those bottles, and actually it's not just compressed air, but sometimes they include some kind of a substance, which kind of propels it out. And that's something what I heard you should not use. And the reason is because as the solvent, as this gas comes out, it expands very quickly, and it also causes a rapid cooling. And apparently what I heard, that's not always good for the optics. If you basically cause the lenses to shrink so quickly because it's so cold all of a sudden. So what you should use is you should only really use compressed air, and not those air blowers for in those bottles. Because it's not just compressed air apparently. But it contains some other stuff as well. So what I've made before is actually made myself some kind of a compressed air blower by using a bicycle pump and pumping a plastic drinking water bottle. And then on the top, you got to check one of my videos. I got this. I put a handle with an air. It's called an air gun. Yeah, looks like an air pistol, and you push it, and then there is a sharp stream of compressed air coming out. It's a homemade solution, which was quite cheap, and that's what I'm using to clean my microscopes. Okay, but this one is an electrical air blower. Okay, if it's an electrical air blower, then it's like, yeah, then I think you're just using the air, the ambient air. Okay. Hot blow dryer could cause alignment issues. I would not use any temperature things. Okay. Yeah. So that's simply something. Don't use. I wouldn't use any. Yeah, and I don't think that those hot air blowers actually create a strong enough airstream. I kind of doubt it, because some of the dust can be quite stuck to the parts, and sometimes. Yeah, but what I'm using is simply this here. Simply a brush, yeah, and I'm kind of brushing off, not the objective so much, okay, but the other parts of the microscope, the lamp and so on. The lamp likes to collect a lot of dust, so I use a brush to kind of clean it off. Okay, you know what? Actually, I did promise you to put something under the microscope today after all, because I've been now talking again over an hour already. Okay. So you know what? Why not give it a little try just for the fun of it? And let's use this one over here. Okay, and I'm just going to put a water sample under the microscope so that we actually have also looked now at something under the microscope today. Okay, and over here we're going to use this here. I will use one of the big cover glasses, okay, which I have to clean because horrible, really. I'm not happy, and yeah, but that's simply something that I've gotten used to right now. So usually when, of course, after I clean them, also using some hot water and so on, it's actually, they're much cleaner than before. That's gonna be fine now. Yeah, why not try a little bit of this here? So what did I do here? I'm gonna show you. I got this glass over here from an aquarium. Simply some fresh plant material. And there were very few water microorganisms growing on there because obviously the fish, they scraped off everything from the surface of the leaves. And so there was actually not a lot to be seen. And I took a little bit of the plant material, put it into a separate container and added a cereal grain. Yeah, just one cereal grain. And then I waited a week and it started to turn a little bit bad and ugly. And that's a good sign, okay? And there are now basically, yeah, I just go in here and I take some of the material out here and put it on here. But usually what I also do, and this is where I just want to show you where I use those tweezers, what you can do as well is you simply go in here, you take some of the solid material and put it in here as well, because usually there are lots of microorganisms then growing and sitting on on the solid material because it's a food source. So, and I don't know where I put now I just had my little cover glass which I just cleaned and I don't know where I put it, so I'm just gonna take another one. Yep, I don't know where I put it. So here it is. I put it here right next to it. Okay, so here it is. And let's give it a try. Up. And yeah, I'll put it now under the microscope because otherwise we haven't seen anything today under the microscope, and I still want to show you a few things here. Why does this not work? Because, of course it doesn't work, because you have to turn on the microscope. How is that? Okay. So, okay, and let's put it in here. And let's see if we're a little bit lucky. Yeah, I mean, it's of course way too bright. Okay, that's the 4 times objective. Let's go a little bit higher here. And this is the wrong filter setting. And we can already see a few nice ciliates here. And you know what? We're gonna change the background. You know, that's the blue background that some of you have been wondering about. Okay, that's basically a setting of the prism of my microscope. You know what? We can put this here better. And yeah, and that's basically the result after approximately 2 weeks. Yeah, after I added the cereal grain, and then you're able to see a lot of life. Okay, and one of the things that I started to appear now in the last couple of days are rotifers. Just want to show you here as well. Where is the arrow? Arrow over here. Here's the arrow. Okay, see this one over here? That's a rotifer. Now they also started to appear. So generally, what you're able to see is you're able to see that over time, yeah, there's a progression of different microorganisms. Okay, so sometimes yeah, certain microorganisms become dominant, and then after a couple of days, different organisms become dominant. And here, now that's basically a rotifer. They started to appear now a few days ago. So let's go up a little bit with the magnification again. Okay, we need a little bit more light. Okay. And this is also basically the place when you look hard enough, you might be also lucky to find some amoeba. Okay, but again, because they kind of move very slowly, they're kind of difficult to see. Okay, so I'm going to quickly go through some of the questions here. So the ear alone wouldn't be able to get out many of the things of your objectives. Okay, how do you keep your working environment clean so that nothing gets on your samples? I mean, things like hair, they drive me crazy. Okay, I will tell you, dust is generally a big problem. Seriously, I don't know what to say. Keep your microscope covered, vacuum clean everything. It is a problem, what shall I say? On the other hand, if you see some occasional hair or dirt or dust on the microscope slide, I don't worry so much because the natural sample often contains even more. Okay, so yeah, so in that sense, yeah, unless the dirt is too much, yeah, it shouldn't. Yeah, it's got to be tolerable, right? Yeah, yeah. Here that's a paramecium. My last question makes sense in extreme macro when you see the result after stacking is over, some between 200 and 300 images. So if I find something there, I need to repeat everything. Ah, I get it. Yep. Okay. Dog poo for the first time, lots of worms and stuff. Just make sure that you're careful with the infectious material. Okay. Because of course the parasites, bacteria and so on. Okay. And yeah. What's cereal? Yeah, oats. So basically a wheat grain or yeah, exactly. Some kind of starchy material. And if you've got a wheat grain and if you compress it between 2 spoons, okay, then you can, yeah, you can use it, or in breakfast cereals. Not cornflakes. It might also work, but if you use kind of cereal, it's kind of this flat oval thing, yeah, and just put one of them in there, and then you wait approximately a week, and it's going to be full. Don't overfeed. That's important. Yeah. What specimens do you recommend for observing for beginners, and what is your favorite specimens? For beginners, I recommend that you start honestly, yeah, practicing the standard stuff. Try putting vegetables under the microscope, onion skin, for example. Okay. This is a standard thing. Your hair, everything that's sufficiently small, thin, and transparent will work. And I would say that many people, of course, are very fascinated by those living, moving creatures that you see over here. That's why I'm showing them as well. But again, to the density and concentration that you see them over here, you will rarely find them in nature like this. You have to really wait until the plant material starts to decompose and break down, and then you're able to see more of those, or add a little bit of food. Okay, so that's basically, yeah, yeah. I see lots of stuff here. because they're all feeding. They're of course all feeding on the decomposing plant material. In the background, what you see, these might be bacteria, or most certainly are bacteria. Okay, all of those dots, and those ciliates and flagellates and whatever you see over here, they will actually feed on the bacteria. Okay. What is this here? Ah, there's another one. Okay. So, Happy New Year from Iran. Happy New Year. What are these smaller round microorganisms you see on the slide? I mean, there's so many of those. Identifying them just microscopically, because the biodiversity is so high, is a little difficult. But generally, those larger ones that you see over here, these are not bacteria, obviously, because they're too big. Either ciliates or flagellates, depending on whether they have many little hair around them, then they are ciliates, or if they have one long extended flagellum, then they're flagellates. Yeah? And what is this? Yeah, this one over here is a paramecium, of course. Yeah. Single-celled, the favorite ones because they're quite large. Yeah. Thanks for answering the question. Anyway, I really love your channel and feel lucky I found it. The amount of information. Well, thank you, of course, also. You know what I'm going to do? I'm going to quickly zoom in a little bit more. Up. Oh, now I bumped it. Now I bumped the stage. Oh, a little bit. Here we are again. A little bit more light. And a little bit more light. And I have the camera I can adjust as well to make it a little bit brighter. Everything I have to refocus. Yeah. And one of the nice things that you see here is of course the water stream that those little creatures are creating. Okay, that's one of the nice things. Yeah, I just wanted to show those to you as well, because after all, I did not only want to show you home lab things, but what I would like to ask you, if you want to also send me more pictures of your own home lab for me to share, or if you have any other pictures that you want me to share, please send them to me, Oliver@microbehunter.com. And in the next live stream I will share them. Just a short piece of information. Next Saturday I also want to do another live stream, but it will be a little bit earlier than today. The reason is because I have an appointment, and there I've got some time conflict, and I'd like to do it a little bit earlier, so maybe around 6 hours earlier than today. And so I will just want to inform you on this issue. I don't know yet which topic I should talk about, so this is always a little bit of an issue that I have to think of an interesting topic. But if you have any recommendations, then of course I'm willing to accept those. Okay, and you can send them to me as well. Okay, this seems to be like a gigantic air bubble here. Okay, sometimes what you're able to see is that sometimes the microbes, they will actually gather around an air bubble, because this is where the oxygen concentration is the highest. Yeah. The magnification here is already, I'm using now the 40 times objective. Okay. Okay. Yeah. Interestingly, not so many. I wanted to actually find some Vorticella. Last time there were quite a few Vorticella, but I'm not able to see them anymore. Yeah. But as I mentioned, sometimes there is a progression of different microorganisms over the course of several days. Okay, so what I'm going to do now, folks, is I'm going to leave it at that today, because it's been now also again over 1 hour. I wish you, in any case, all the best. I want to wish you, all of you, a happy new year. If you have any further additional questions about any equipment or so, please post it somewhere, write me an email. I will basically pick up on that, and I'll include it also in the live stream a little bit. I hope it was interesting again for you. Please, again, if you want me to share a few pictures that you send them to me. I also will try, if you subscribe to the newsletter, I want to keep you also updated a little bit on all of the videos that I made in the previous month. So if you want to subscribe to the newsletter, there's a link below. for those of you who are new to the channel or were new to the channel, this is only one of my 2 microscopy channels that I have. Please also check out the other one, the main channel, so to say, if you're interested, where I post also videos once a week about. Well, I wish you all the best for 2023. See you again. Yeah, I wish you all the best. Happy microbe hunting as always, and bye bye, all the best. And enjoy the New Year's party if you celebrate one. Bye bye.